Hey everyone, I'm Joe, your instructor, and welcome to Microbiology 205. And this is your recording for Lesson 1, An Invisible World. So I'm going to give you a brief overview of the main points of this lesson and then talk a little bit about the application of the understanding coming out of this specific lesson. So we're going to start with what are microbes, and then at the end, we're going to cover why they really, really matter. So what is a microbe? Micro, meaning invisible, organism, meaning alive and an organism, however you want to define that. So microbes are organisms that are too small to be seen with the naked eye. They are found absolutely everywhere. They're in the soil, they're in the water, they're in the air, even just floating around. Obviously, they're going to be on your body, in your body, just absolutely everywhere you could think of, there is microorganisms living. Now, there's a few everyday impacts of microorganisms that I think really illustrate how important they are to our daily lives. The first, which is covered in this lesson, is yeast fermentation. So, if you've ever baked bread, you take your water, your flour, and then you add yeast. Well, why would you add a microorganism to your bread? It's because the yeast actually will eat up carbohydrates in the bread and then produce carbon dioxide, and that carbon dioxide bubbles up and makes the bread rise. So if you've ever proved bread, that just means you let it rise over time. That is the yeast eating something in the bread and producing carbon dioxide to give you these air bubbles. So if you have yeast in your bread, and of course let them do their thing for a long enough time, it's going to be this nice, light, aerated bread that is what you typically see. And then if you don't have yeast, that's going to turn into something that's like a flat, dense, almost just pure crust. And that is, of course, probably not, depending on what you're doing, as joyous to eat as a nice piece of bread. Of course, microorganisms are in a bunch of different things in our daily lives. So, yogurt and cheese, for instance, are made through bacterial fermentation. And I'm going to use this word fermentation a lot throughout the course. That just means that you put a thing with a microorganism and let the microorganism act on it over time. There is a better definition, but for just the general understanding, that is what fermentation is going to be. It's going to take and break down parts of the additive. The microorganism is breaking down parts of the additive and then changing it into something else. Alcohol, so beer and wine and ciders, those are all fermented. Again, that's generally going to be yeast. So we use yeast in a ton of things. Yeast are absolutely fantastic microorganisms that are all around us. And I think this illustrates really well that not all microorganisms cause disease. I said first, they're in the soil, they're in the air, they're in the water, they're all around you. Well, why aren't we sick all the time then? And there's a lot of reasons for that. A lot of it is our own immune systems protecting us, a lot of it is general cleanliness. But the other thing is that a lot of those microorganisms, they don't care about us. They hang out in our environment and we might use them for things, but they're not going to impact us in any major way. And some, in fact, most of the ones that do impact us actually impact us positively. So you have microorganisms living on your skin. That's really important for your skin health. You've probably heard about the microbiome. So that's all the microbes that live inside your body, generally in your gut. So the gut microbiome is what that is generally referring to. And those are really important for your gut health, but then also your brain health and your general health overall. These microorganisms, we give them a place to live, and they give us the benefit of protecting us from all those other microorganisms that are trying to do us harm. So if you think about the plague, for instance, it's a terrible bacteria and it infects and killed a lot of people in the ye olde times, many, many times over, because there was plagues and then there was plagues and then there was plagues. So the Black Death is the most famous, but there was plagues due to that specific bacteria previously. And then later, we actually in Arizona are a reservoir for plagues. So don't go out playing with prairie dogs or anything like that, or you might end up with the plague, actually. So that's a little fun fact just for living in Arizona. That's something you could have to worry about, but don't. It's not that big of an issue. Uh, but where I'm going with this is that the bacteria that do infect us are actually in a smaller category than the things that support us, and a much, much smaller category by magnitudes from all the microorganisms that are around. So the microorganisms, so that includes bacteria, that includes archaea, that includes other things that we're going to get to. A lot of those do infect us, but a lot more we live in harmony with, and a lot more we just don't even fall into their purview of what they can interact with. So that is a little bit about why they matter in everyday life. Now, I'm going to talk a little bit about the history of major events in microbiology. So, we have all of these microorganisms around us. Well, what did people think before modern science? And what people thought of was this spontaneous generation. So, you would have a sewer, it smells terrible. You have a bunch of garbage. It smells terrible. You have dead bodies or corpses of animals, the throwing out of a butcher shop, let's say. And that is all going to be something that we as people naturally reject because it smells bad, it looks awful, and we know that people get diseases when they're around those things, when they're interacting with this. And that gave rise to the spontaneous generation theory that there's a miasma around these different areas that are associated with disease. So it just spontaneously happens. The flies around the sewer spontaneously generate out of the sewage itself. And then the diseases associated with that spontaneously happen because there's a miasma around what is there. And you can't really blame people for thinking this because microorganisms are, by definition, invisible to the naked eye. So they're seeing the results, but can't identify anything other than, well, these things that are awful to be around, they must have something innate about them. And they do. They have microorganisms in them that are going to impact us in a native way. And that's where those specific microorganisms are breeding. So they came up with this philosophy of miasma, of spontaneous generation, and that was commonly held belief until Antony von Leeuwenhoek developed the first ever microscope. Now, the reason he developed this in the first place was actually for industrial application. So, if you're a trader, if you're a merchant, and someone says, "Oh, I have the best rugs in the business, I have the best silks, I have them at 1,000 count per square inch," let's say, well, that's really small. I can't really count that with my naked eye. Do I trust you or do I look at it under what is a microscope and count them myself? So that was the original purpose behind the development of the microscope. But then he saw microorganisms and wasn't able to explain it. This is a guy who's not really in it for discovering microorganisms. It's a byproduct. And that happens a lot in science. There's these things we see that don't make sense. And that is a byproduct discovery of something else we were doing. And this is the case. But then people picked that up and started doing actual in-depth scientific research on microorganisms. So Louis Pasteur, he's the one that disproved spontaneous generation, that theory that there's a miasma and things just come out of filth. And of course, pasteurization, Louis Pasteur. So he developed pasteurization, which is really important for our everyday lives. If you think about milk, it comes out of a cow's udders. It is not, even in modern times, the cleanest thing in the world. We have to treat that milk if it's going to be able to last a long time. So he invented pasteurization. That's where you essentially you boil. It's not quite a boil. It's just you heat up everything until all the microorganisms are killed, and then you make sure through a couple different methods that you're not reintroducing microorganisms before you seal whatever the product is in a container. Pasteurization is used every single day in 1,000 different products that we consume all the time, and it keeps us relatively very safe. Now, if you crack open your milk and leave it out for a day in the Phoenix heat, that is not going to help you. The Arizona heat will make things grow in it rapidly, even though it's been pasteurized. So uh, it's a it's a system that has helped a lot, especially in food science, for making it so we have long shelf lives on different products. Now, Louis Pasteur also advanced germ theory. So that's the theory that no, it's not a miasma, it's actually these microorganisms that are causing disease. But at this point, we didn't really know if it was all microorganisms cause disease or specific microorganisms. And that's where Robert Koch came in. So he created Koch's postulates and showed that specific microorganisms are what actually are leading to diseases. So now we know, oh, it's not all microorganisms, it's specific ones. It's the ones that are dangerous to us that we should be worried about. And the culmination of all this is we have a much better understanding of infection. We started developing aseptic techniques. So if you've ever watched a show that has a surgery, they're all gowned up, they all have gloves on, everything's tied off, they have face masks on, and that's to not protect them from the patients, to protect the patient from them, to make sure no one coughs and spreads a microbe into a open wound, because then that patient might get an infection. Things like hand washing, disinfectants, vaccines, uh, antibiotics, this was all developed off of this early understanding of microbiology. But we're getting into a point where I'm saying, okay, we have these specific microorganisms. Well, what do we do with that information? How do we actually categorize these? And that's where taxonomy comes in. So this is how we name and classify microorganisms. It is simply the science of naming and classifying things, is taxonomy. Uh, for taxonomy in general, it's hierarchical. So it's bigger buckets going into smaller buckets. And it starts at the top with a domain, and then a kingdom, a phylum, a class, an order, a family, a genus, a species. We usually care most about species, genus, and family because that's something we can actually work with in science. Species mainly are what we're going to be working with and focusing on. But when we talk about domains, that very, very top, it's broken into three different domains. All life on Earth falls into one of these three domains. No matter how weird it is, it fits into one of these. That's bacteria, which are prokaryotes. So these are unicellular organisms that are very small, non-nucleated. We'll talk about that later in the course, but that just means that it doesn't have a nucleus. Archaea, the same, prokaryotes, so no nucleus, and these are often extremophiles. If you look at bacteria and archaea, just as they are, they're both prokaryotes. They're both these unicellular organisms, for the most part, that are going to be acting fairly similar. So the only way to tell them apart is by looking at their genetics, their different types of proteins they produce, the cell walls that they have. And from that, we can pull and parse these into specific lower orders on that taxonomic scale. And then, of course, there's eukarya or eukaryotes. These are often multicellular, but not always multicellular. So you can have unicellular eukaryotes, you can have multicellular eukaryotes, but what classifies them specifically is that they have a nucleus within them. So they have all the outer machinery, and then in the middle, they have a nucleus. And that is how they're going to be able to be told apart from archaea and bacteria. Now, eukarya is very wide spanning. It includes protists, fungi, plants, animals, parasites. So it is a large bucket with a lot of diverse things in it. Why is taxonomy important? Well, it lets us, first off, ensure that we're talking about the same thing. If I'm a scientist talking to a scientist in Germany, as long as I'm using the proper terms for a species, we know we're talking about the same thing. And then it informs treatments. If you have someone who is infected with something and they have a runny nose and a fever and a cough and chills, Well, you might think, oh, that sounds like flu, but until you know for sure, you don't know how to treat them in a clinical setting. So it might be the flu, it might be a sinus infection that's a bacteria, it might be a weird parasite they picked up on vacation to Uruguay, for instance. So there's all these things that really feed into ultimately the treatment of patients. And that all comes back to knowing what we're talking about. And that requires taxonomy. Now, all of these different organisms vary a lot in size, and we're going to start talking here about the fact that while these organisms fall into different buckets, a lot of times, like I already mentioned with bacteria and archaea, they can be hard to tell apart. So when we're looking at the size of the organisms, a virus, which I'm touching on viruses for the first time, we'll talk about them more as the course goes on, but they're very, very small, around 10 times smaller than bacteria. And this is because they don't package everything required for life themselves. They have to rely on another cell, whether it's a bacteria, archaea, or eukaryote, for replication and for their life cycle. So they can be very, very small. Then bacteria, they're also very, very small, oftentimes smaller than a human cell by about 10 times, but there's outliers. Same with archaea. You can have really big bacteria, you can have smaller bacteria. So it depends on what exactly you're working at. Again, that is why taxonomy is important. When we look at human cells, they're about 10 times bigger than bacterial cells. And then when we're looking at plant cells, they can be oftentimes bigger than human cells. But there's overlaps during the entire course. When we're looking at different things, it is important to realize that they fall into these major buckets, but also it's a Venn diagram. They're going to overlap sometime in some way. And that is why knowing what exactly we're talking about becomes very, very important. So we're going to talk about the five types of microorganisms and their driving characteristics. So first, there's bacteria. We've talked about bacteria a lot. These are single-celled prokaryotes, and they often have a diverse metabolism. What does that mean? So they can take in a lot of different what we call metabolites, and that just means it's sugars, or they're able to photosynthesize. So they're taking in the rays of the sun, the actual protons from the sun, and converting that into energy. They're eating, if it's not sugars, maybe they rely mostly on proteins, so they eat meat mainly. Maybe they can actually survive in really extreme conditions. They're called extremophiles when they do this. So maybe they're actually able to live off of heat from hydrothermal vents. That's another way that these bacteria can live. So they have a very diverse metabolism, extremely diverse domain bacteria are in. Then there's archaea. These are also prokaryotes. Sometimes they're not single cellular. A lot of times they are, sometimes they're not. And these are often extremophiles. So they're going to be feeding off of heat, salts, acids, bases, things that bacteria normally would not consume is where archaea find their niche, where they like to live. But of course, there's going to be overlap there with bacteria and also some eukaryotes. So fungi, they're eukaryotes. These include yeast. Yeast is unicellular. So you might start thinking, well, prokaryotes are unicellular. All eukaryotes are multicellular. And that is typically true, but yeast, they're unicellular. So again, there's that overlap and why taxonomy becomes so important. Fungi also include molds. So if you've ever had not so clean shower and lived in a very humid area of the world, you might get this kind of orangish film. That is a type of fungal mat, actually. So there are these multicellular fungi that can live in a bunch of different places. There's protozoa. These are unicellular eukaryotes that are motile for the most part. Again, I say for the most part. You're going to get that a lot. So there's buckets that things fit in, but there's also these overlaps outside of those buckets that you're going to see. So protozoa, unicellular eukaryotes, motile. Modal means that they're able to move around under their own power. Some bacteria, archaea, other organisms, microorganisms can't move. They don't have machinery to actually allow them to move. Protozoa generally always do. There's algae, so these are eukaryotes that are photosynthetic. They're key in aquatic ecosystems. So you have algal mats that bloom in the ocean. Algal blooms is usually how you'll hear them referred to in the news. They'll bloom. They take up a bunch of photons from the sun, turn that into energy, divide and divide, bloom up, get eaten, and then go back into this aquatic ecosystem of they bloom, they feed fish and other microorganisms, and then they kind of go away for a bit, and then they bloom back up. Hematoliths are parasitic worms. So these are important because they're studied a lot in microbiology due to both their life cycle. So they have a lot of microscopic stages during their life cycle and then a huge impact on disease. These fall into eukaryotes. So they're large multicellular eukaryotic organisms that are responsible for disease and often studied. And then, of course, there are viruses. Viruses, you'll notice, were not in any of the three main domains of life, the domains being bacteria, archaea, and eukaryotes. The reason for this is they are acellular. They don't have the ability to replicate by themselves. They're what we call obligate parasites. They have to have a host to survive and replicate and mutate, so they evolve. They need that host cell. This is why viruses can be very dangerous because they require us. So they want to infect us. They're going to figure out new and better ways to infect us as they evolve, just because of the pressure that they want to always replicate. Now, this is different than parasitic worms. Parasitic worms can survive on their own. They'll wait and in their life cycle go through different stages where they're not even parasitic. They're developing and then they become parasitic in a certain stage and then they infect and then they use us as a host to replicate in. So if you're a fan of the Alien movies, that would be a parasite. The xenomorph would be a parasite versus a virus that is required to live in a host. Now, I know this is probably a lot of information, a bit confusing, but just focus on the fact that these are the main types of microorganisms we're going to be covering and talking about. Familiarize yourself over the course and keep in mind that there are those buckets that they fit in, but they also will overlap in certain ways. And that is why knowing the exact organisms we're talking about and that taxonomic hierarchy is so important. In closing, I'm going to talk a little bit about wider applications of microorganisms. So, one of the main things is the production of biologic drugs. So, biologic drugs are drugs that are proteins or enzymes or peptides, things like that. And a lot of times, microorganisms are used to produce those, whether it's yeast or bacteria or eukaryotes. So, in my personal research, we spent a lot of time developing a system and applying this system to the production of biologic drugs. So what we did is take a microorganism that already infects plants. So if you've ever been hiking and seen a tree with a big bulbous growth, that is a microorganism, a bacteria specifically, that has infected the tree and overtaken its cells, saying, produce sugar. I want sugar. I want to eat sugar. Produce me sugar. And then the tree does that and grows these bulbous growths on it. That bacteria, we took the machinery out of that hijacks the plant cell, used that for our own applications, and applied it to other types of plants. So we are able to make things like vaccines and immunotherapies and enzymes. And for my master's degree, I actually made spider silk in plants using that microorganisms machinery. So that's a really cool application of taking something that life has developed itself and making it work for us. Yeast and the making of bread and alcohol and a bunch of other different fermented food items is kind of the same thing. We're just taking a microorganism that already has all the machinery we need to do what we want it to do. And there's the application there. It's a huge thing in food science. Now, when you're going through the course, You're going to probably have a lot of questions. I want to encourage you to go ahead and if you're ever confused about anything, if something's not making sense, message me and I'll do my best to clear it up. And I hope that this introduction and lesson is a fantastic first step into the world of microbiology. Have a great day.